All right, we'll go through a really quick, just PowerPoint, what we're going to be doing, key concepts in both linkage mapping, QTL mapping, GWAS, and then we're going to go through an example. So, general steps of linkage mapping. So after you get your data from, if it's GBS data, it would be out of Chris's pipeline, or in the future, some of these linkage mapping software on the polyploid level will be able to take what MCHAP can output. Right now, I don't think that probably can do multi-allelic yet. So we're going to do some filtering, then we're going to estimate the recombination fractions between the markers, and then grouping markers, ordering markers. Those are the general steps of linkage mapping. So for filtering data, typically in an experiment, a lot of times you have off types, or you have duplicates, or you have things that just don't belong. And this is just an example of, so the mapping populations I talked about at RGC for black splice or COSPR, all those, this is the raw data when I got it back. And what you see here is a principal component analysis of the genotypic data. And what you see is a triangle. On the triangle, what you see up here, let me see, where's the pointer? Laser pointer. Up here is one parent, and it looks like that's stormy weather. Here's another parent. I can't make out what that is. Oh, that one's my girl. And this one down here is bright eyes. So you have the two big populations that I talked about, where we found the QTLs and stuff. But what you see is there's some off types that don't belong. There's a guy that's over here, that's over here, and a group over here, and off groups. So at first, when I tried to do my linkage mapping, a lot of the things didn't make sense. My groupings didn't make sense because I had not run a principal component analysis yet. So once I took all that into account, got rid of this guy, got rid of this guy, got rid of these, and these numbers are supposed to be individuals, but they clustered more closely to the parents, so they might have been selves. So on this slide, this is an old slide, so some things that can cause bad data are selves, off types. So in the greenhouses, a lot of times you'll mislabel plants, you'll maybe collect DNA from the wrong plant. For us, roses are sometimes budded, but sometimes you'll accidentally collect the rootstock. So that's happened to us before. Other things of bad data, let me think. I can't think of anything else. All right. So then, filtering data, so off types, missing data. So we'll filter on missing data on an individual level. So a lot of the genotypic files that you'll get will have rows of markers, and then they'll have columns of individuals most of the time. Some programs it's reversed, but we'll do two levels of filtering, one on the individual level. So looking at individuals, seeing how much missing data, you could set 5%, 10%, 25%. It's kind of arbitrary, depending on how good your marker data is. And on a, you could do it on an individual level, looking at all the markers, or you could do the marker level, looking across individuals. And you can also look at the segregation, expected segregation ratios, and look for segregation distortion and filter based off that. So then the next step would be estimating recombination fractions. And we saw plots like this earlier, Chris was showing us. And this estimates the distance of markers from each other. And in the blue are markers that are far apart. So there is recombination. The markers that are in red are really close to each other, so they segregate together, and there's high linkage, so there's very low recombination. And you will rarely see plots like this if you use GBS data. not that clear. This is from array data. That's why it's such a clear red line. This is of roses. So you can see these 7 groups. This is linkage group 1, linkage group 2, 3, 4, 5, 6, and 7. So estimating recombination fraction matrices in autopolyploids, there are different approaches. So the 2 main software that is part of tools for polyploids, MapPoly and PolyMapR. With PolyMapR, what the strategy they take is they first look at all the simplex nullplex markers and estimate recombination fractions with those and build a scaffold. And from the scaffold, you can get a chromosome level and you can get homolog levels. And after you build that scaffold, chromosome level and homolog level scaffold, then you estimate the recombination fraction of all the other marker types to the simplex nullplex types and fit those markers. So computationally, this is a more less intensive way to get your recombination fractions. Now, MapPoly kind of takes another approach. They kind of just say, okay, let's just calculate all the pairwise between all marker types. So if you are working on an institution that does not have high performance, then you may have to do a little more tinkering and use PolyMapR, because you could typically run that on a laptop, 16 gigs of RAM, and typically be fine. MapPoly, if you have a very filtered data set, you might could run that on a PC, but most of the time you would need a high performance cluster, just because of this step is very heavy, because pairwise recombination fraction estimates as you add markers is exponential. Then you're going to group your markers into chromosomes. So using both software, you're going to have to do some kind of grouping. And like I talked about earlier, PolyMapR kind of at the beginning, you have to make a scaffold and then you fit everything to it. MapPoly, you can use UPGMA, and that is just a clustering algorithm. Or you can use the genomic data. If you have a good physical map, or you have a good annotated genome, and it matches it's very good for your species and very close to your crosses, your biparental crosses, you can use that. Or you can use a combination of both, and we'll kind of, in the code, go through that, all 3 methods, and kind of look at that. And for the ease of today's demonstration, PolyMapR is more hands-on and not as automatic. So we'll use some functions in PolyMapR, but we won't actually construct maps in PolyMapR today. So after grouping your markers, so after you're getting your markers into your linkage groups, then you need to order it. And there's 2 kind of, the 2 programs kind of use different methodologies. So PolyMapR uses MDSMap, which is basically looking at taking the information of your markers and their recombination fractions and kind of almost plotting it on a 2D. It's kind of like a PCA, but it's MDS, multi-dimensional scaling. So it puts it into an order on a 2D or a 3D plane, and then estimates which ones are closer to each other, and then it orders the markers. However, when you use MDSMap, you don't get as good collinearity to your reference genome. It'll be a little more scattered. MapPoly uses the hidden Markov model chain on a predetermined order. So in MapPoly, you can either use the genomic order from your genome, or you can use MDS. You can run MDS on all the markers and then order the markers that way. So what MapPoly does, it takes the dosage information and then it takes some kind of order and it combines those 2 into a hidden Markov chain to combine that information to give you the most likely phase of each individual and the most likely map. So for the tutorial today, you probably will, most people have their own laptops here. Between 4 to 8 cores, 8 to 16 gigs of RAM. It might run on less. It might run on 4 gigs of RAM. We'll just see if your computer crashes. It shouldn't. Have matplotlib installed, and we will download the scripts and required data. I'll go through all that in a second. So for QTL mapping, after you do your linkage mapping, what you're going to have is something like this. For every single individual that you will have genotypic probabilities and homolog probabilities. So we saw some pictures of this earlier during RGC during a couple of talks, but what this is this is the map position down here in centimorgans, and on the y-axis is the probability that this individual has each one of these parental homologs. So A, B, C, D is parent 1, E, F, G, H is parent 2, and at any given location, you're only going to see a summation of 1. Summation of 4, because it's each one is at a probability of 0 to 1. So you'll see 4 homologs at any given position. What you see here are where the recombination breaks are. And in this example, the D homolog didn't have any recombination on it. It inherited that entire homolog. Then you're going to add information. Basically, this is a big data frame just visualized. The big data frame has every individual, every linkage group, every map position, and every homolog. So if you think of that as it's a huge data frame. It takes that and then takes your phenotype files, and then it kind of if we simplify it, it basically does some sort of like regression to see which homolog is responsible for the QTLs. That's a very oversimplification. So the method that QTL poly runs is REMIM. That stands for estimated multiple interval mapping method. I can't remember off the top of my head. Yes, I have to look it up. But what it is it's a model that starts with a no QTL. It does a forward search of QTL at a very relaxed p-value. Then in model optimization, it uses a more stringent p-value to either keep a QTL, and then during the optimization the position slightly changes, or if at this more stringent p-value the QTL doesn't meet the p-value, it gets kicked out. And these steps keep going over and over and over again until no QTLs are added or no positions change. And at the very end, there's QTL profiling where the score statistics are updated conditional to the final QTLs at the end. And then we'll kind of just hit GWAS as a really quick explainer, and then we'll go to the hands-on portion. So key steps in GWAS, or for GWAS poly, is you just read in the data, you set the population structure, and you'll filter that. You can filter the data from minor allele frequency, and then you just run the GWAS and it prints out the results. That's it. So let's get to the tutorial, because we'll probably get stuck, and most of the time of getting stuck is probably installation if y'all haven't installed the programs and getting all the stuff that we need downloaded. So first, we need to go to my GitHub. So just go to GitHub. See if I need to make this larger, don't I? Did somebody paste that into the?
Yep.
I'm going to mapping example. you're going to click this code button, and you're going to download the zip. All right, everybody caught up?
It's a little bit slow.
Yeah, it's going to be a little slow because everybody's accessing at the same time.
Yeah.
I tried to take out all the large files, all the files that we'll be generating. I'm trying to take all of those out. So. It's just github.com/Jeekin Lau, and then mapping example should be pinned at the top. Presentation for all this stuff fast. Yeah. Then once we start doing it, it's like so hiccups. Yeah, especially when we start.
Yeah, it was just slow.
Yeah.
Are you the Teams?
What? Yeah.
I have shared them. Yeah, don't.
I probably should have put it on the flash drive. That's not very high marks. Not very high security. Cybersecurity.
Oh, yeah. Yeah, he get really using. We just get how.
So?
Without the mechanism. So that's the link to, yeah, we can go.
So link up.
So we can probably just check in. Right. Everybody?
Well,
Could you get it? It's weird. And then example. Can you just go to GitHub? Yeah.
Yeah.
Is
is it just really slow?
Yeah, it's really slow and he doesn't have access to Teams. Just like. And then
Pressed it.
and yes, it's already. It's yeah. Okay, let's start with.
Are the people online having problems too? I don't think so. So if you have it downloaded, unzip the file. So right click and unzip it. And inside that folder, you'll eventually get to the tutorial.
No, don't worry about this yet. It would be on your local computer wherever you've done.
Oh.
Yeah.
To the tutorial. So if you want to follow along, the entire RMD is already compiled in the HTML, and this gives you all of the output that we're going to generate. So the HTML is there. Or I would open this, the mapping example RMD. And it'll look something like this. It's just a R notebook, markdown notebook.
Can you just zoom in?
Yeah. View, zoom in. Is that zoomed in enough for online? Probably.
Yeah.
For online? Yeah. Not good enough.
Yes. And you can just open HTML. One more?
One more? View, zoom in. Okay. So make sure you're down here. Make sure it says that it is inside the tutorial folder, and to make sure you can go up here to session, set working directory, and to source file location. So again, session, set working directory, to source file location. Once we have that, everything should be good. Okay, if you don't want your little R markdown to look like this and look like code, what you can do is click visual, and it'll render it to more readable, and it'll show you the boxes of what to execute. So hopefully, most people have map poly, polymap R, QTL poly, and view poly installed for later. The more experimental versions of the software would be installed using DevTools and install GitHub. And just a quick introduction while we're trying to get everything done. So what we're going to use today is a very clean data set from a SNP array data, and it's actually going to show some of the data that we talked about in RGC 11 about the black spot Cercospora and defoliation. We'll have all those traits in here. This is just one of the populations on a really stripped down SNP array data set. So we've done all of this. We've done all of this. So you can always go back to this RMD and find the links to the more in-depth tutorials from the developers. So from Marcelo, his vignettes, he has 2 vignettes on the tetraploid and on a hexaploid. QTL poly, there's a new vignette from Gabriel, and there's an old one from Guy.
We don't want to click on the zip file.
View poly, Chris has her tutorial there, and polymap R has their tutorial up there.
Since we just need to open this file in Studio.
So really quickly, while we are still kind of waiting for people to get their stuff.
Is there like a right-click option to open?
Stuff right. This is what your data will look like. I'll open it up in Excel.
So, Studio.
So before we go into map poly, your data will look something like this.
I mean, it's harm document. Yeah, I learned it.
What you'll have is on each row, you have a new marker. On the first two columns after the marker, you'll have P1 and P2. You'll then have your linkage group and your position. These two are actually optional, but if you have it, it's highly recommended that you have it, because if you have this, this probably means that you have a well annotated genome.
Is there an unset option?
And then you'll have all the dosages of all the individuals after. So this is what the data looks like that we're about to read in. So the rest, once we get everybody situated, the rest will be just point and click, point and click. So the rest will be fast. Anybody still have problems? Still have problems? It won't extract.
No, it's extracted.
Not
Have you
set.
opened it? Yes.
Oh, set the working directory for it. It's not set right now. Or just go session.
The file new image.
Set working directory.
This is what the image.
Just source file.
Oh, okay,
It's
that's
just-
good, right?
That's good.
It's
It's already there.
Yeah.
All right, we can get started.
You know what you do? I've got no image.
All right, I will need you to actually comment out this line, or else it's gonna mess up. So on this line, get rid of this line, or else it's gonna set your working directory to something else. So put a hashtag in front of it to comment it out, or get rid of it. Everybody commented out this line?
Yeah.
Okay, if you've commented it out, then you can press play, and it will import the data. When you press play and it imports the data, it tells you a little bit about the data set. It says it's read in this following data. It knows that the ploidy is 4. There's 169 individuals. There's only 2,075 markers, and all the markers are informative. So in this case, it's a very clean data set. It's not going to be 100% informative on a real data set. And they can also input VCFs. This isn't actually part of the tutorial, but this is just showing you it can take a VCF, an example VCF. It takes a little longer, but I'll show it since we're still having troubles importing the real data.
Oh, yeah.
So we can also we also can import VCFs, and then this is the data structure that you'll see once you import the data, the genotypic data into MapPoly. So it tells you things like ploidy, number of individuals, number of markers. It gives you have all the individuals' names. You have the marker names. You have the dosage of the parent 1, parent 2. You have the information that we read in on that Excel line. These two. That becomes this part. chromosome and genome position. If you have a VCF, you'll have a sequence reference and an alternative allele. And if you have GBS data, or if you have any sequence-based data, you'll have the marker depths, probabilities, and then you'll have a thing called the genome dose. And this is the genome dose is just from here on to the end of the file. All right, that just shows what's read in. Are people still having a problem installing? That's what I thought. That probably takes a long time to install. Does it install faster if you tell it not to compile? Maybe?
That's a good question. I think, yeah, by default you say yes, but it just that it's going to take your packages you need to compile because it has the later version. Yeah,
I'll just wait. We'll just wait.
Okay.
I've run the software before.
All right, is everybody else fine in the room? All right, let's go. So we'll do some quick QC. And what we're going to using for QC is PolyMapR because it has some good, basically, it has good tools to visualize what's wrong in the data set. So what we're going to do is we're going to load PolyMapR. We're then going to, so inside of MapPoly, there's a function called export data to PolyMapR. And then we're going to export it to a dosage matrix. So if you put your cursor at the end of the line and just go Control Enter, it will only run that one line. So that's loading in PolyMapR. You can run this line. What it does is it gives you the input for PolyMapR. That second line, this one. And what we'll do is then we will run a PCA progeny, which is a function inside of PolyMapR that lets you run a simple PCA. And what you see is something like this. And what you see here is there's this main group, and there's also this side little group. And let me open this up so that you might see it better. So you'll see a few things that are wrong. You see some overlapping things that kind of look like bolded, and they're really close to each other. And you see these off groups. What you should see is that P1 and P2 are actually in the middle of this cluster, but you can add another thing to PCA progeny, and I think it's called highlight C P1 P2, and let's color them red. Let's run it again. What? Oh, red. I need two reds. C, red, close parentheses.
Yeah, the paper, I guess, is like checking this out.
Okay.
So what you should see, if there was no contaminants and if there was nothing wrong, what you would see is this cloud of individuals, which are your progeny, and your P1 and P2 should be pretty close to the origin. But in this case, there are contaminants, and so the P1 and P2 are clustering a little further away. But what we see here are this guy up here. I don't know if you can see the cursor. For those in the room, I'll point to it. But this individual looks folded, that individual looks folded, and then there's off types. And what I did was I purposefully introduced duplicates. So that's what those are.
I did, and so I just.
All right, so if you looked really closely, you could find the contaminants, and these are the names of the contaminants. So let's label those as contaminants. So now this is a vector called contaminants. Let's see if you look at this is just a vector of all those individuals that we had listed earlier. And then we can do something else. Polymapper allows us to do something else called screen for duplicate individuals. We run this chunk. It does a piercing correlation. Where's it not displayed? It does a piercing correlation of the genotypes, and everything up here, you can set your Pearson correlation. You can say, oh, if it's 95% similar, it's the same individual. If it's 90%, it's the same individual. In this case, I think we set the screening at 95%. So any individual that's 95% similar, it will be called a duplicate and collapsed. So now that we know what the duplicates are, the printout tells us which individuals are. This is wrong. I don't know why it says that. F1 and all these. That's not correct. Let me see why. Oh, this is what's wrong. I put in the wrong. I put in the wrong matrix. So on this line, on the screen data line, the dosage matrix needs to be polymap R dosage matrix, not all matrix. All dosage, not all dosage. So change that, and then it should tell you what it will print off another one. So it should look like this. Different than earlier. And it'll tell you which ones are the contaminants and which individuals were removed. So using that information, we now know what the duplicates are. So we now can make a vector of duplicates. So now we have two vectors. We have contaminants, these are these, and we have duplicates. Are these. So what we'll do is we'll take those out of the data set. We'll combine those two into a new vector called remove. And these few lines will remove remove those individuals from the genotype data. And we'll write that to a new file.
Did you know the contaminants from looking at the PCA?
Yes,
Yeah.
so you can, from the PCA and from looking at the duplicates. So the PCA stuff was that one, I guess I could rerun this side. Rerun this, it'll. It was these individuals were the contaminants. And then this one tells me which ones are the duplicates, and the printout tells me who the duplicates are. So that's a polymap R good functions that are that I've been using to kind of screen my genotypic file before I go into mapping. All right. Now let's read it back in, the genotype back in. We need to comment this guy out because this is not my working directory now. So comment this first one out. And we had made this new genotype file where we removed after QC earlier. So it should read in. And now you should only have 157 individuals. Before we had like 169 or something like that. Then we're going to filter the data. We're going to filter the data by, I don't know why these X's are here. I don't know. It should still run. So we're going to filter it by the marker. So we're now back in map poly. These are map poly functions. And we are using the input data. We input that new data that only has 150 something individuals. We're going to filter by the marker. We'll have a filter threshold of about 10%. And then we're going to do the same thing by individual with the filtering missing threshold at 10%. Because this is a very clearing data set, you're not going to really see anything removed because it's so clean. Now, there are no rules for what level to use. So typically in an array data set, in multiple array data sets, I have been able to get good maps with 1% missing data, 5% missing data, or 10% missing data. Now, if you are using 1% missing data, you're very stringent and you're only keeping the really, really good markers. If you're using 10%, you're allowing a lot more markers. So then now your chain's going to take longer to run. Your recombination fraction matrix is going to take a lot longer to run. So you have to balance how much missing data you want to give it and balance your computational power. So let's just run this box. It runs. And it spits out a few things. It tells us how many individuals there are and the frequency of the different marker types, but that's not important. Then we're going to filter by segregation distortion. So a lot of the times, after you filter based off of missing data, there still are a lot of markers that don't fit Mendelian segregation distortion. Now, there's pros and cons of using this because if you're working in a population that has known segregation distortion, then maybe you don't want to leave those markers out, right? Or maybe you do. I don't know. That's all depends on you. But here we use a simple Bonferroni corrected p-value. So our p-value is 5%. We're going to divide that by the number of markers. So remember earlier in DAT, the data, we have the number of markers. So that's what we're saying. We're saying 0.05 divided by the number of markers, and that gives us our new p-value. We're going to run this map poly function called filter segregation to get the marker names that need to be filtered, and we're going to make a sequence. So let's just run this box. This box gives us a lot of pretty outputs. You might not get this color scheme because this color scheme is quite new. This is in a newer under development version of map poly. You might get different colors. I'm not sure if you're using the CRAN version. But there's other printouts. There's printouts of, let me see if it's here. I think it's here too. No, it's not here. It's printed out to here. You get printouts like you run sit in this one, the sequence. It tells you how many markers are in each linkage group based off genome information. You have the number of markers that are there, so on and so forth. So this section, I'll go ahead and run. Go ahead and press this because it will take a couple minutes. This is calculating the recombination fractions. Now, I think I forgot to mention before you click it. Oops. I don't know how many CPUs you have. So what you can do, you should be able in your console type parallel, detect cores, and then enter, and it'll tell you how many cores you have. Most of you will have between 8 to 16. Most people. So if you have 8, for instance, don't set it more than 7, or else your computer is going to kind of be really slow. So always leave at least one core available. So go ahead and set that. If you have 4 cores, set it at 3. So on and so forth, and you can start that. Let it go. And then we'll go try to figure out any other issues. Yeah, I think yours is fine. Yeah, it should just, it takes a long while to calculate. That's good, that's good. That's right.
So I need to change it? No?
No, you just let it run, just let it run.
Oh, it's running now?
It's running.
What's the percentage of turn the computer?
Yeah, it takes away a while to run. Yeah, if it's saying that, it's not.
Just you type something. Good. I don't need to know how many cores I have. Oh, so I thought microwave was ready. I have CPUs.
Or this one that you need to pass because you're saying. So maybe you want to
Because that's the only program.
Yeah, yeah.
Single command.
I can see how many it has.
So I usually press that. But otherwise,
You have 8.
so once.
So for this one.
Now you copy the information. And for
So there's actually 2 steps that takes a long time. This takes a long time, and the HMM chain takes a long time. Now, there are ways around this. So if your data set is too large and your cluster is running out of memory, there's another strategy where you don't do pairwise comparisons against all markers. You can only do it within chromosome. If you're going to align using your genome position anyways, what you can do is go ahead and separate out all your markers based off chromosome, and there's easy scripts. Just email me. I have the scripts. I use them all the time. And then you just do it by chromosome. It just lowers the amount of calculations a lot.
It's also the same if you compare as
By all?
only and
So if you do, it depends on your use case. If you want to cluster using a clustering algorithm like traditional, so people like coming from JoinMap, they're going to do against all markers and then do your clustering using the recombination information. If you want to do that, you have to do all of them at one time. But if your pipeline that you have a good reference genome and you're going to use the reference genome order to use in your HMM chain anyways, then there's no really reason to compare a marker on 1 to 6 or 1 to 4. It doesn't really make any sense. So mine says it was done in 107 seconds. It doesn't show up there very well. Maybe I need to.
You are?
No, is that better?
Yeah.
Yeah, that's better.
Good.
So 200 seconds, not too bad.
Oh, you are?
But I have had this run for days.
Oh, when you had a lot of markers.
And before I was like, hey, I'm using the genome order anyways, so why am I comparing one linkage group to another linkage group's recombination fraction? It doesn't make any sense. So if you get done with that portion, you could probably go on to the next step. The next step, all it does is it takes this big recombination fraction list, it makes it a matrix, and it plots the matrix, which is what we showed earlier, the big red line going through. And then you get this pretty thing.
Thank you.
Just fresh.
A crashed computer?
Yes.
Oops.
Yeah. Some computers, it's not understanding how to just speaking this.
How many cores does she have? 8?
They have enough cores, but it's not like.
It's not going?
Oh.
Yeah.
I did it, everything.
My matrix looks better than yours.
Really?
Yeah.
How?
Just
Oh,
a
same data set.
better color contrast. Yeah. That helps.
Good way to test the reproducibility. Different results.
Oh, yeah.
So for the next step, it's visualize the combination matrix. That's it. Think a little bit of visualize.
So if you didn't have a reference genome and you did everything, just run the next block of code, and what it would do is it would take basically what we just plotted, the recombination fraction matrix, and it tries to cluster the most similar ones together using a UPGMA algorithm. In this case, because it's super clean, you get your 7 groups. It's never this clean with real data. So you may have to tinker quite a bit.
Oh, those are your 7 linkage groups. You just.
We just trust the Rose genome. So we use we cluster by the genome order. And
you often encounter, like if you actually check and see if you occasionally get linkage across context according to the root.
we do have that in our diploids.
We've seen some pseudo linkages. Sometimes.
Yeah.
And we don't know why. Probably because
It's probably statistical.
Yeah.
Small population size, a lot of markers, some of them look like they're linked there.
And then you just go in and say, this is linkage group.
Yeah.
Mm-hmm.
Yeah, there's 7 linkages.
So one of the outputs that it gives you.
Reciprocal translocation is what happens, the data really.
Let me see.
These guys do.
Yeah, this is similar the output.
Yeah.
It's the no cost, it's virtual.
Yeah. That's good.
Comes together in a real way. So that's what you think.
Now seems that we are.
I don't know.
It's a 674, 521, Use that one.
I don't know. That's a good question. But I think the numbers on there are what the genome order is. because 2 is our largest chromosome in rows, 3 is the smallest one, and that seems to be.
Okay.
I think it's just maybe how the. I have no idea. I think it has to do with where the breaks start breaking off is what the orders are. Because I think UPGMA is also used to cluster things like genetic distances and stuff like that for building like evolutionary trees and stuff like that.
Okay.
I rarely ever use this, so that's why I'm not super. I've never gotten it to work well using it by, at least not in GBS data. In array data, it falls out pretty good. So what we're going to do next, there's 3 things. So before we go into mapping, we're going to make lists. And inside of these lists, it's going to be a list that contains however many chromosomes you have. So for roses, we have 7. So it's going to make a list of 7 other lists. And inside of these lists for each chromosome, it's going to have a sequence. So a sequence in a particular order. And then it's going to have the recombination information that we had earlier. So that's the 2 things that we need for mapping. We need some kind of sequence, at least for HMM chain mapping. We need a sequence and we need the recombination information. So if you run this chunk, it runs pretty fast. It's making 3 big lists. And what it's doing is it's making a list here of both the genomic information and the UPGMA, the intersect of the 2, just the genomic information, and just the UPGMA information. And at the very end, we'll compare the number of markers in each group. And this code is from Gabriel, who now the maintainer of QTL Poly. I just never took it out of the tutorial, so it's in here.
I trust him, so I just press one.
Yeah, I trust him, so yeah, yeah.
The problem is,
And what we see, if we, it's a little messy up here, but if you type COMP down here, what you'll see is, if you look at just the genomic information, typically they'll have higher amounts. So they don't match up perfectly. So UPGMA, you might have chromosome markers that belong to other chromosomes that are going to other chromosomes. If you just look at genomic, it's only going to be what's in your genome order. And if you look at the intersect, it will only take the intersect of those 2.
Those numbers are the number of markers on each linkage group?
On each linkage group using each of these clustering methods.
Oh, okay.
So either clustering using just the recombination information, just the genomic information, and the intersect of the 2.
Okay.
So just for today, you can go back and try to do them separately and see how it affects a map. But today we're going to just take this first one. So we're going to make a new object, LGS, and assign it the LGS inter list that we had made earlier. So we'll do that. And when I talk about list inside of list, this is what I'm talking about. We opened up the list. This is the list, or we can go up to enter. This is a list entered that we now made LGS. Inside of this list, you have things. You have 7 chromosomes. Inside of this list, you have a sequence. Inside the sequence, you basically have a sequence of markers in genomic order. And then inside the 2-point, the map-poly 2-point object is basically an object that contains all the 2-point recombination fraction information. And that's what's needed to do the mapping. And then we will go to the next one because it takes a little bit of time. Let's just do 1 chromosome to show you what the chain looks like. So if you click this next box, it starts running down here. And this is the chain running. And it runs really fast right now because it's a clean data set. On a real data set, it's going to take a little bit of a while.
But this is real data set.
This is a real data set, but a clean real data set.
It's real data set.
What I did was I went to the maps that we have constructed for our papers, and I went into an existing map and took like every 10th marker. So I know they already map well, and then I just take all the markers. So what this is doing is adding markers, adding markers, adding markers. Sometimes if it doesn't fit properly, or if it doesn't fit right, it'll kick the marker off. But I don't think I've seen any being kicked off, maybe. And inside of the chain, there's a lot of functions. You give an input sequence up here, which is your list, LG1, and then SEQ. You have a starting set, so that's just how many markers do you want to start the chain with. In his example, sometimes he uses 3, sometimes he uses 5. You can play around with it. There's no set rules. These thresholds are just thresholds that he uses. And then there's just different parts of the chain if you want to like delve in deeper to what each thing means. In his vignette that I pointed to earlier at the very beginning of this, he explains every single thing, what each thing is. One of the important things is this last argument, the sub map size difference limit. So during a HMM chain, it starts out with a 3 and then it starts adding markers. But every time it adds a new marker, it re-estimates a new linkage map. And this submap difference is how many centimorgans after. So it has an old map. So let's say it starts with three markers. Then when it gets to 4 markers, it has the map size of the old map and the map size of the new map. And if the difference between the two is more than this guy, the marker gets kicked off and it goes on to the next marker. So if you set this last argument, sub map size difference limit, really high, then that's like, it'll keep all the markers. But you'll have large gaps in the map. So you have to kind of juggle that. So what we saw in here is that we mapped 99.6% of the markers, all but one. So if we scroll through here, we can probably find which marker got kicked off. And I don't see it. Oh, at the very beginning, it couldn't find. So it started 1, 2, 3, didn't work. So they went to 2, 3, 4 to find something that made sense to start the chain. So it threw off the very first marker. Now, let's close this portion. So we're going to now do, if you click this next button, it's going to do all 7 chromosomes at a time. Now, the problem is, does anybody have less than 8 cores in here? If you have less than 8 cores in here, down here, these 3 portions, change this to.
Oh, I see.
Yeah, whereas 7, make it
3.
less. Yeah, 3 for you. So it's just 2 lines. Otherwise, you can just run it and it should work. So when I run go, it will run. But then it doesn't do any output like earlier, so you don't know how far it's been running. It'll just tell you, oh, true, 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 and then it'll just sit there and run. Now, a little trick you can do is you can go into here, this tutorial, and there's these output files. So I'm telling R to write into the output files what is in the console of each of those chains. And if you're on Windows, you can open terminal. I know what it is in Linux, but I don't know what it is in Windows. I think it's tail. No, tail is in Linux, right?
Yeah.
PowerShell will work too.
PowerShell is tail also? Maybe. Stop picking it up. Maybe. No, it doesn't have tail. It's get content. is in PowerShell. Get content, let's say linkage group 3. What? Doesn't exist. Oh, the name's wrong. Output. Then you can see it running. Then you know what percentage it's at. So if you're using PowerShell, it is get content and then the name of the file and then dash weight. If you are using Linux or Mac, you could use tail dot minus F, yeah, and then the name. So that's just this is useful on high performance just because if you're using a full data set, this could take a couple days and to kind of just check in to see which chromosomes are done, which one you're waiting for.
Thinking, yeah, we've already done the HLA chain. We'll do the competition.
This is representing the two. This is the two parental maps, and this would be the integrated map. But this is basically the 4 homologs coming from one parent, 4 homologs coming from the other parent. It's just a visualization. You don't really. I don't really use this that much. It's just pretty nice. It's a good visual, and in her tools, it uses the object that you used to print this, and you can zoom into like kind of haplotype areas that you need, like we get to that level.
Yeah.
Because our QTL that we don't have big enough populations, we can't really make any good inferences on, oh, this is a good haplotype, but that's what this is for.
Okay.
Okay. Yes.
You're just getting the get content.
Get
out,
content,
and I
yeah.
couldn't remember where we're
Get
going.
content, and then get. That means this is on you. What's the what's the meaning of this?
It's running. Oh, there. Yeah, it's gonna take a while.
Over PowerShell window here. One, no, actually, 2 is the longest. Or 2. Oh. That's right. But I don't think you have a version of PowerShell. That's why this is not
Crazy.
Yeah. It doesn't run
Old worker, this is very better.
You just need to look at the. You just need to look at the percentage.
Yeah. There's enough, just.
With the click the file, you are run the environment.
And because linkage group number 2 is the largest, I'm just going to keep checking
Yeah,
that.
we are trying to translate the language system.
Yeah.
That's at 40%. So when 2 is done, the rest of them will probably be done. I think 2 is big, and then 5 maybe.
It cannot print dots or bars.
Yeah.
So the dots are-
Yeah.
No graphics.
But you see in this example, a lot of things are being left out. Not included, not included. The chain couldn't fit those. And another interesting thing is, or maybe annoying thing is, the longer the chain gets, the more markers that are added, the longer it takes to add the next marker. Because it's looking back through the rest to make sure to basically find the best fit. So I have had data sets take longer than the HPC at Texas A&M allows a job to go.
Sorry.
So you may have to email your HPC person, be like, hey, I need more than 15 days. Give me another 10 days on this job number. I've done that before. But typically what happens is on those really long jobs, more often than not on the newer clusters, they go through maintenance in between my jobs and then you're just like, I can't do anything about it. You have to start it over. Maybe I should upload this file to the GitHub and then just download the file. And then we can keep going instead of mapping the rest of it, because I thought this was going to take faster. Maybe it's already there. Let me see. The map's error in here. No, it's not, because I told it not to go. Give me a second. I will fix that issue.
Yeah, I know. Yeah.
Yeah, pick up. All the backups.
Yeah.
All right, since it's taking so long, if you go back to the GitHub, I uploaded the resulting file. If you download this data, this object.
Oh, that's just to see it?
Yeah, well, let's just skip this portion.
Yeah.
Because linkage group 2 is taking too long. So I'll click here. And then I will just download this. So go get this maps error data, click on it, click download, and place this R data into the working directory. Or you could just click on it. It should bring it into the R.
Oh, can you just show us again where?
Yes. So I'm going to stop this map construction first because it's taking too long. So let's go here. Go back to the GitHub page. I just uploaded it on the main page. Refresh it. This maps R data. Click on it. And then download it. And when you download it, just click it down here. And it should open it. It says, do you want to load this into your global environment? Say yes. Now it's there. And then we can skip that. We can skip onto here.
Oh, yeah. This is the one that's really hard and sticking on there. So
So what you can do after everybody imported this thing, this maps error from the GitHub. There's a few commands you could use. Down here in the console, type summary _ maps and maps, this maps error object. Enter. Let me run it again just to be able to see it all. There's your map statistics. So if we would have run everything, this is what we would have gotten. You have your maximum gaps over here. You have total amount of markers. What type of markers? You have how many markers per centimorgan here. In the length of each. If you said plot map list, and you say maps, what you get is a graphical representation of your maps. They look all right. They're all at about less than 100 centimorgans. You can get another output called plot. If you just say plot and you say maps error, and then you look at the first level for chromosome 1. So square bracket 1. You'll get that thing. There are 2 parental maps. And it's just telling you parent 1, A, B, C, D, and this is the A or the B, because the SNP data is either A or B allele. This would be a lot more colorful if it's GBS data. Yes.
Did you run map, the one that created map before? Did it work for you and you only gave us the map.error?
I got maps.error.
Yeah, and what about maps?
Oh, that's a good question.
You did run it on your session.
I didn't save it.
Okay, yeah.
But we only need maps.error, I think.
Okay, yeah.
We can skip that portion. Yep. So after you run the maps, what you do is you'll apply a 5%, that's a good catch. You apply a genome-wide error that you want. So if you have just the maps, it's going to be a lot larger because the HMM chain is not allowing as much genotypic error. But then when you run, let me see if I have the 2 plots that we saw earlier in Chris's session. When you allow more error, if you have, let's say 5%, for instance, then you allow the chain to make more changes in the genotypic dosage calls all the way up at the raw data area. And when it does that, then it changes more. So on the left of this, there was only less than 0.07%. I think that was at HMM, just the maps. And then maps at 5% is almost 0.5% of genotypes are changed. So I think we don't need, yes.
Do you have to set a constant error rate for every genotype, or is there a way that you can read it? Quality scores for the genotypes.
Map poly can use whatever the error scores in the VCF if you choose to do that.
It's usually using a global number.
Yeah. I was just wondering.
Yep. So we're gonna skip the genotypic. We're gonna skip. Wait, not where do we skip? Oh, because we skipped this at the end. This also makes the error maps error. So we're back to here. We're back to here. The genome. Now this last one is also something important to kind of look at. How do I get to, oh, here. If you run this line, plot genome versus map. So this last line here, what that will give you is a plot of your genetic map versus the physical map. Now, what you want to see, what you don't want to see, is any big gaps along this. So sometimes if the data isn't good, or something like you'll see the genetic map start here, but then this is the x-axis is the physical map, and the y-axis is the genetic map. So sometimes you'll see it start like the red dot starting here, and then that would mean your chain missed like a lot of physical area. But it looks like these cover pretty well. There's a little bit of gap here on 5. And there's a little bit of loss of recombination on 3, which is known in literature.
Very short arm.
Yeah. Other than that, you can kind of use this graph as a diagnostic. What you don't want to see, I don't know if I have a picture of it.
Which one?
I have a picture of it. Because Tessa, one of the graduate students in our lab, was rerunning something. She's probably listening right now. Let me see if I can find the picture that she sent me so I can show you a problem. Maybe not. I can't find it. Yeah, I can't find it. The problem would be if this guy was shifted over. If you can think of if the red dot started here and all the way down here. So if you shifted this over, then it would mean that you missed a lot of physical area. And sorry, I don't have a good example here because these are good maps.
So now if you can split it into the same.
Yes, because there's a microcentric cost. Probably happens that.
So before we go to QTL mapping, we need to, we now have a map, we have phased maps, and we need to estimate the genotypic probabilities of each individual. So remember we saw earlier in the presentation here, this guy? Well, we need to estimate this for every single chromosome, every single position, and every single marker location. So you run this next block, and it will estimate those. And we're going to save that to a thing called genoprob error. So it's running. And it takes a lot longer on bigger data sets, but this is relatively small. And then when you're done with that, to visualize some of this stuff, what you can do is you can calculate the homolog probability. So this is another function inside MapPoly that calculates each individual's homolog probability off of this genotypic probability object. And what that gives you is, takes a while, a really large data frame. I'll show you what that data frame looks like. It should pull up without crashing my computer. So you see how this similar thing that we got earlier, right here? This is generated from a large data frame called homolog probs. We come over here into the environment. Homolog probs is here. Inside there's another homoprob data frame. You open that. This is what the data frame looks like. So for every marker, every homolog is listed there, every individual, and the probability that any one of these is the right one that's inherited for every map position. So this is a huge thing. But that is the data frame that is used to make this figure. Okay, we talked about that. We talked about that. We talked about that. All right. I don't know if there's a break. I'll keep going.
3 p.m.
3 p.m. Oh, let's keep going. Let's do the QTL poly part. So I don't know if people have QTL poly installed. You'll know real quick if you run that line and it says no, not installed, but it should be there.
I got it. It's running. Chrome.
So typically whenever you're done doing your mapping takes a while, you're going to get this genotypic probability file. Save that, put it away, don't rerun your maps. I would save, whenever you're done with your mapping, I would save a few objects. So I would do this, I would say save, and I would typically save the DAT, probably your maps, maps error. If you had the regular maps, I would save that also. Those are probably the most important things to save. So you don't have, and there's relatively small data objects. So this guy should already be in our environment, but if it's not, we can read it in. We're going to read in a phenotypic file, and that phenotypic file looks like this. It's going to have genotype in the first column, then your traits. And so let's go ahead and run this first step. So what it's doing is taking the genotypic probability object, and then it's reading in a phenotypic file, and it's combining the two. So read data is a QTL poly function that reads in all of this and makes a data object that's just a big list. And what it read in was that we have Podia 4, 157 individuals, 7 linkage groups. And right here, the step size is typically people just put it at 1 centimorgan. Could put it at half, but if you put it at half, it's going to increase your computation a lot more. Could put it at 0.1 if you wanted to, but your QTL scans will take forever. So that is how often you want to, down the map, you want to sample for QTLs. And this step, for every new map, you will need to do something. So Gabriel's working on an improved method of this and a faster method, but this is how we do it now. You simulate 1,000 fake QTL, and you calculating the null model for those 1,000 fake QTL according to your phased map. So for every new phased map, you're going to have to run this, and it can take half a day to run or something like that. But I've already ran it on our maps that are good here. So let's not run any of this, but we will read it in. But before we read it in, this is what it's doing. Remember earlier in the presentation, we talked about REMIM, so that multiple interval mapping method that has a no QTL model, then adds one and it takes away one, adds one, takes away one, kind of in multiple steps, forward and backward elimination. So this is the null model. And normally you would not actually run all these steps. You would just run one function and it would do all this for you. But they wrote this in a way so that it could be kind of didactic and so that you could go through and you can kind of mess with the models yourself if you wanted to. So I'm saying I want to look at the phenotypic column 1 in our phenotype file, we have like 5 traits, but I'm just going to look at one just for speed. And of course, after the null model, no QTL model, you have no QTL. Makes sense. Then you're going to do a forward search. So forward search, you search for QTL, your data is the data, model is you have to use the null model to now search for QTL. The size width 15 is the width of your window that you're searching. Forward, backward. Oh wait, forward, you only have forward right now. And then number of clusters, you can change this to 3 if you don't have 8. And yeah, let's run it. And it's gonna say I have like 2 or 3 QTL, I can't remember. So while it's running, it tells you, it prints out what's happening. So right now it's found a QTL on linkage group 3 at 49 centimorgans. And it's still going, it's still adding. Yeah, this is the forward search portion. So during the forward search, it takes the null model, so no QTL model, and then it looks for the most significant one. So it adds that one first. Then it goes and takes into account that first one, and it looks for the next one. And then it's looking for the next one. And then after it does all that, then we have to go backward. So we use a more stringent one, more stringent p-value, and then we start trying to take away. But that's.
So Jeekin, every time QTL is found, it uses a cofactor for the subsequent search.
I think so.
That way it accounts for QTL in the background, right?
But I don't know why this is taking so long. Shouldn't take so long.
Yeah, because for QTL, then you transfer
If your forward search is done, go ahead and start doing the optimization step. So if we go back to here, the forward search searches for a relaxed p-value. In optimization, things can change.
Yeah, exactly. Yeah.
Yes.
To the top, something that you say is RGS.
Which one? That's it. RGS.
This one.
Yes.
Do you want to explain it to me?
It should be. Let me see if it's in. GeoPro, where is it? And not. Oh, it's. I think it's a really big file.
Oh, okay.
It's 77. But it should calculate pretty fast if we have.
Yeah.
Is not calculated?
Very interesting for some people who fail to pay.
If they take the object, this is a small object, the map there,
Yeah, okay.
and start from there,
Yeah,
that should be fast.
Thanks.
Yep.
So this one came across without any combination. One these are the two that came from the combination. These are the three classes. This one we have the two chromosomes that expect
Okay, so
That's the sorry, I'm looking for
The data there.
so that you can say cut.
If you're done with the forward search, you can go ahead and do the optimized QTL portion. And see down here, when it's optimizing, it's telling you what it's doing. It's refining the QTL positions. These positions are the marker numbers. Then it's excluding the non-significant ones. So using a more stringent p-value, it's kicking out ones that didn't meet that. Then it's refining the QTL positions earlier that it kept, and it keeps going back and forth until none are, no more QTLs are dropped or positions change. And what you should get is a after optimization, you profile the QTL, which that just calculates things like heritability, which is QTL probably uses the word heritability for the QTL as like a R squared. So it calculates that. It calculates the top and the bottom, confidence intervals.
This option has the probability for each one of those. So it's connects my whole
After it's done doing the print profile, I actually copy this and put it in the terminal because, or in the console, because for some reason it doesn't like to print correctly in our markdown. I don't think I can see what it does. Yeah, it looks all weird.
Yeah, so now I'm just generating that and using the radius.
You just get a lot of statistics of what QTLs are left, the upper and lower bounds. And then at the end, this is what you get, like a plot. Plotting of the profile. So this is just looking at one trait, looking at black spot, and we have two QTL. If in plot profile, you tell the data, so the data that we imported at the very beginning, the model, which is at the end, the profile mod, and then if you say SUPINT, that's like superimposing the confidence intervals, it will shade where the confidence intervals are. And the little triangles down here are the QTL peaks. So what we should do is we should run all of the traits. And remember earlier we talked about that score null object that would take a really long time to calculate, the 1,000 fake phenotypes that you kind of get a null for all of it to get a genome-wide significance. So basically, it's placing fake QTL all through the genome. It's calculating the null score, so like the null model for all of those, and then it estimates the genome-wide like null for all the traits. So I calculated that earlier, and you should just be able to run that first line. So just click this button, and it should read it in. You'll get an error if it's not there. And what this does is it's going to start running the QTL analysis for all the traits in that phenotypic file. And I think we can take a break while it's running.
Hello,
All right.
we are back.
We are back. So after running this REMIM, which is, so you don't have to do all of the score null forward search back automatically, just run this one line. What it'll give you in the output is what it's doing in each step. So for instance, here, this is a forward search. It's looking for the QTL, and no more QTLs are found. Maybe a putative will be found, but it didn't make the significance. Then it refines it, and then it does backward elimination. And then it keeps doing rounds and rounds and rounds and tells you how long each trait takes. When it's done, that's this line. The next line, plot profile, gives you this, what we saw earlier. The shading is the confidence interval, like we described earlier. But this can be sometimes a little bit kind of crowded. So then if you say grid true, this third line, you can separate out your traits. Or if you want to look at it another way, you look at it as fourth line, and then you get it like this. Where the black bars are the QTL peaks, and the colored bars are the confidence intervals. And then it's color coded. So the next step would be fitting the QTLs, and what this does is it gives you the kind of the statistics. It runs really fast, and this was the part that didn't go right over here. So you need to copy all of this, and then run it in the console. And what it will give you is a list, all the traits. Trait 1 black spot, where the QTLs are, the variances of the genotypic variance. This heritability is equivalent to like a R squared. It's the QTL's heritability. Then we can look at effects. If you run this next line, what this shows us is the effect of each QTL. So if we look at this, black spot QTL1, which was on linkage group 3, we see that P2 allele F lowers the phenotypic, I guess, mean. So if you're an individual that carries the second P2 homolog, then you're going to have a lower black spot. Conversely, if you have homolog H, you're going to have a little higher. So you have that for each trait. Just going through. You can look at those. Just look. What you notice here is most of these resistance traits, a black spot here, Cercospora here, defoliation. A lot of these are seems to be primarily simplex phenolplex traits, primarily. So you have one homolog that's contributing a lot to the phenotypic mean. When you get into more quantitative traits, there's a PhD student in our lab, Amrit. She's looking at flower coloring traits. Oh, these plots are so messy. You have no idea which homolog is responsible.
That means there's more than one
Yeah.
homolog. And
They look more like this, where you're like, okay, so is it this guy or is it this guy? Like what? So it's not as easy to stack QTL. So when it's a simplex phenolplex trait, it's very easy. I want this homolog, I want this homolog, I want this homolog. And Chris will kind of show us how to do that in View Poly later. So at the end of this QTL session, what we need to do is we need to save the maps, the data, the REMIM mod, the estimated effects, and fitted mod. And what I will do is I'll run that. Oh, I can't find it?
No, mine can't find it either.
Uh-oh. Maps error. That's a good question. I don't know what's wrong.
Is the path correct?
The path is correct. Name tutorial. Oh, because I don't have a. How do you make a directory in R? Like, what's the equivalent of mkdir in R? Who's Olivia? What's make dirt?
Yeah, I'm searching.
Just try and make it.
Oh, I'll just make it here.
Maybe you can just make it. Yeah.
So inside a tutorial, oh, just go to here. Go to the file place, go to mapping example, make a new folder here, folder, and name it, what did I name it? View Poly, View Poly _ files. Yes. Okay.
In R, it's dir.create.
Oh man, I made it in the wrong place. Oh, yeah. dir correct. So dir
Dot
create
yeah.
a path would be dot and then view poly _ files and all in. I need that.
And you forgot the O in poly.
Oh, there. No, wrong place. P, not ploy. Oh, the joys of live coding. All right, there. Now it should work. Now it's going to save all those things that we just made. You can go and make sure inside of your View Poly files that you have 5 files. And one of the reasons I didn't want to upload all that into the GitHub was because there's one file that's pretty big. It'll take even longer to download. So I think everybody should have 5 files. All right. That's that portion. Now we need to look at the GWAS. What you can look at is inside of the tutorial folder where we were at, open up the GWAS RMD. It should be here. And we are going to start, comment out this first line.
Yes.
And then you can run the first line, but make sure you comment out the line because you still want to be in this tutorial folder. So in this case, we are using a more complete genotypic file because when I use that subsetted file, you don't see the peaks that you should see. Just because in GWAS it's single marker like analysis basically. And all we're doing here is loading in GWAS poly. We're loading in the GWAS file. We're loading in the phenotypic file. We're just loading in the names. And then this last read GWAS poly, we'll read it all in. One thing to keep into mind is the number of traits that are in your file. It's important for you to put that here, because GWAS poly allows you to have cofactors. So maybe you have 2 locations. Maybe you have some other cofactor you want to take into account. The number of traits will tell the program which columns are traits and which columns are cofactors. In our example here, in our phenotypic file, we only have blues. So it's already, all the cofactors are already taken into account. So you just read it in. So is everybody back to the GWAS polys section? Yeah. Maybe no. Are we still trying to save the QTL poly?
Yeah.
Okay. Oh wait. So once the data is loaded in, we can actually talk about it. So once the data is in, there you have to account for population structure. So we just took the same data set from the biparental mapping population. You wouldn't do this on a normal GWAS, but you could, like, just to see if you have QTLs, like, really quickly. You normally do this on a diversity panel, and then you have to take into account how closely things are related. And in the older versions of GWAS poly, what they did was they had a covariance matrix, a K matrix, and basically used all the markers to kind of build this matrix. However, the new method, it uses, so for each chromosome, it builds a matrix. And it, like, say for instance, if it's working on chromosome 1, it will leave out all the chromosome 1 markers and use all the other ones to build the covariance matrix for chromosome 1. So it's local method means leave one chromosome out. So there's 2 methods there. You can run it if you want. So that's important. The other thing that's important when you're running a real GWAS and not just in a biparental mapping population is your minor allele frequency. And they just suggest that you set it at 1 minus 5 divided by n. Mathematically, I don't know why, but I just take his word for it. And then we can I'll just let this run. These will take a little bit. Oh wait, I didn't run this.
Oh yeah. Okay. So it's a very random.
So you've downloaded the view from
Here you have to.
So then after you take into account the K matrix or the local method, you can run your GWAS. It's running right now. It's running on 1 trait because all the traits take a long time. When you have 30,000 something markers, it's making single marker. Basically, it's running single marker tests, basically. So it's almost, with very simplified, is basically run a simple ANOVA on every single marker. But with other things in, it's taking into account other things. So it's running. It should be done soon.
And so it's also like Emma or something.
Like what?
So it's also using linear model using.
Basically, I think in the background it is. You can. Their paper describes all the math, but I don't understand the math. So in the models, they have particular, like, submodels almost. So you can say, okay, I want to run a. Oh, where did it go? Where did it go? Oh, I went to the wrong thing. Oh, here. You can run additive model, a one allele dominance model, a 2 allele dominance model, general models, and I think in their description here, they explain what they all are. So I think I can bring that up. QTL GWAS poly.
Actually, that should have been step 0.
I don't know if he explains it all, but they explain it all in reference file.
I didn't know it was kicked out.
It's my bad. I should have checked.
No, it's not.
So it says here, the additive model is the additive model. That doesn't take much explanation. The X dominance can be any integer between 1 and ploidy divided by 2. So tetraploid, you can only go up to a 2 dominance. Hexaploid, you can go up to a 3. The diplogeneral means that all heterozygotes have the same effect. Diploid additive effects, all heterozygotes have the same effect, constrained to be the halfway between the homozygous effects and general. So there's a bunch of different models. I typically just run it all and let it output that. And I think it's done. And you can get your QQ plots really quick. I'll go ahead and run this one too. And your Manhattan plots. So you get your QQ plots really quick. So from here, we're going to see peaks on 3. We're going to see peaks on 5. Maybe some on 7. Now it's working on the Manhattan plots. The Manhattan plot typically takes the longest to plot. And it takes longer the more models that you include.
We're doing some work together, there's some work. It's fair.
Yeah, exactly.
I'm thinking it's like one day before it's delivered to go make sure that it's not.
Exactly. Laughing at the videos.
Yeah.
Yes.
Perfect.
What you
And
got?
it's, yeah.
What's wrong?
So I just downloaded a new R version.
Oh, yeah.
Because one of the dependencies required like 4.1.
Mm-hmm.
They ran the first. And then I'm good.
Yeah.
I think it was backups.
That's why I use
You might get some errors. Don't worry about the errors. They're still going to print the plot. So this is just the different plots. So this first one is the local method using Bonferroni correction. The second one is using the original method inside of, so using the K-matrix. So things are slightly different. So with the local method, the LG3, it looks more significant than LG5. With the old method, it looks like 5 is more important than 1 on 3. So you'll just have to decide which one you want to use. And then there's also this new other method you can read about in their papers of estimating, of making a correction called m.f. And here's that one. So what we need to do is determine the LD between markers. After you have your peaks, you need to determine the LD between markers so that you can estimate the highest, basically estimate the effect of the QTL. And this one, I don't know why it's running so long. But because this is a mapping population, your LD is really, really high. And the LD should drop off a lot faster on a regular diversity panel. There's your LD plot. Where it drops off. So between 25 million base pairs and 50 million base pairs, just because this is a what's that called? A mapping population. Normally it drops off like here. So using what you found here to see what things are linked, you determine the number of base pairs to get your window. So the highest QTL effect.
Thanks a lot.
Okay,
Yeah.
joining us.
Bye.
Keep in contact.
Yeah, I need to.
And what you get is your 2 QTL peaks. And then you can fit those QTL and get the R squared of those QTL. Now, if you you see how long, how many base pairs it takes for the LD to cut off. Now, if we said in here only, let's take off the 0. And you did this. Now you're going to have a bunch of quote unquote peaks because it's using that sliding window to find the most important snip. So you'll have to play with that to estimate the effect of the highest. And I think that's it. So you need to save this, save for the thing that Chris needs. Save data for. So down here, type save, and then data for, comma, file equals. What did I save it as? GWAS results. GWAS underscore results. Dot R data. Enter. And they should save that.
Yeah, but I'm saying it's used a lot.
It saves it right there. This last line. Or to make it easier, when you to find the files later, you might want to do save you and then add a dot view poly. Or save it there. Oh, it's not. It doesn't exist.
Oh, I just cannot open the connection. Make sure you didn't lose your internet.
It's the name. View poly files. That's what it was. Or run this last line. So it's all in one place. And then now it's Chris and Olivia's turn.